Thanks, Leonore. Can everybody hear me? Mm-hmm. Okay. I wasn't sure with the mask on how well it'll cover, and I'm optimistic that I'll get this to work on Leonore's laptop. It's a PC, and I haven't looked at a PC for several decades. So hopefully I don't press the wrong button. And I did want to do this because when I was first here half a lifetime ago, I didn't really get a chance to talk about the research that was underway in my lab. And this is actually a slide from the talk that I gave on that visit. And it was just a one slide outline of the different types of research that had been underway in my lab for many years. And really, most of our focus has been on fungal morphogenesis. So the focus has always been fungal biology. And a lot of it has been on the growth and development of fungal hyphae. So the mechanisms that underlie polarized hyphal growth. More recently, in a collaboration with two colleagues that are chemical engineers, we've begun to focus on more of a systems level analysis to understanding the signaling pathways that dynamically regulate morphogenesis in response to different types of stress. And the stress that we focus on is cell wall damage. And this has been particularly productive and is it's a project that we're actively engaged in, though most of that work is actually happening right now at University of Nebraska, where I still have students remaining. What I've highlighted in yellow is what I'm going to talk about today, and it's work that we had sort of been tinkering around with in the lab for a couple of years. Then just before, ironically, just before leaving Nebraska, we got a decent sized grant from NASA to work on this, and I left that behind at Nebraska. Then when we moved to Canada, we got support from NSERC and a couple other agencies in Canada to work on this. And so this is really the focus of what most of my students have been working on for the last couple of years. And I'm hoping to build off of this to some of the work that I want to do here. So I've got the slides going backwards. So there we go. So when we talk about extremotolerant fungi, that can refer to a range of different habitats. So perhaps one location that you all have in your house that you can readily identify extremotolerant fungi in is your dishwasher. So several of the fungi that I'm going to talk about today are routinely identified from your dishwasher. It's no fun doing the dishes. I'd rather travel. So being in Canada, we had the advantage of going to a variety of locations and habitats that are characterized by their general extreme nature. So this is one location. This is a beach ridge just outside Churchill, Manitoba. So this is the shoreline of Hudson Bay. The tide is out. And Churchill is up in the northeastern corner of Manitoba. This is a geological paradise. This place is filled with a variety of different rock formations mixed in with biological soil crust. And so we had a field site. It's actually an NGO runs it for us, not far from this location. And it's great to work up here. The problem is 4 months of the year is buried in snow. The next 4 months is filled with mosquitoes the size of birds, and then the following 4 months is filled with hungry polar bears. And for example, when I did this, I had a bear monitor standing next to me with a shotgun. So we're limited to the type of sampling that we can do up here, but there's a variety of environments, and we've done several isolations from there. One of our more productive sites has been a little bit safer is the tablelands in Gros Morne National Park in Newfoundland. So it's right here on the northern peninsula. And this is a unique geological habitat where earth mantle rock is exposed. And so, And so this, all this rock here, it's almost presumably Martian-like. I haven't been to Mars, but it certainly looks like what you would expect to find on Mars. And it's a type of rock called peridotite, and it actually, there's virtually zero plant growth in this habitat. But we've done extensive culture-based and metagenome surveys here and found a number of unique and novel fungi. And in fact, from our metagenomics, this is amplicon-based metagenomics, most of what we find at this site is unknown. So the gray bars here, whether we're counting OTUs or trophic modes, over almost half of what we find, or at least 40%, are not, it's just not that they're unculturable fungi, there's no sequence in the database at all. So this just underscores the diversity of fungi that are present in these extreme habitats that have never, ever been seen before. So the work that I'm going to tell you about mostly focuses not on these sort of rock-infested sites, but instead on sites that are characterized as featuring biological soil crusts. And so, but whether we do the rock surfaces or whether we look at soil crusts, we're really trying to answer two questions by looking at the extremotolerant fungi that are found in these habitats. One is essentially, how do they protect themselves? So on a rock surface exposed to wide daily variations in temperature, wide daily variations in UV exposure and desiccation, what features allow these fungi to adapt and to survive on those locations? And then also, what do they eat? As I showed you at the tableland site, there's a paucity of plant life there, though we do find algae, and that's a clue to the answer to that question. So where we've really focused in trying to answer these questions are on looking at biological soil crusts. Partly, it's just a little bit easier to isolate fungi out of the soil crust than it is out of rocks. The rocks, we need to do a lot of grinding, and that just makes it a little bit more complicated. Nevertheless, what's interesting is we do find the same fungi, whether it's from a rock surface or from a soil crust. So soil crusts are these unique communities, usually consisting of lichens, sometimes a moss, often algae, fungi, and a variety of bacteria. And they form a film, and arguably a biofilm, on the surface of semi-arid or arid soils. And we typically find them, as you might imagine, in desert or Arctic conditions. So we have a long-standing collaboration with Jason Steich's lab at UC Riverside. So they do a lot of their work in the Mojave on soil crust found there. Conversely, we've been looking at soil crust at more northern habitats. So this is, I believe, from the US Southwest. It's not the Mojave, but it's a site nearby. Our work is focused on sites in British Columbia. And the site that the isolates came from that I'll tell you about today is shown here. And so this is in the Rocky Mountain Trench, which in British Columbia, the Rockies basically, you have the western edge of the Rockies, and then you have the Columbia Mountains, and there's about 5 kilometers between them. So this is a deep valley, and this is an old lake bed at the bottom of that valley. So it's then dried up, and these sand dunes are shifting, but they're covered with a crust that largely consists of 5 different lichens and then a mix of other organisms that hasn't been characterized. So with the help of colleagues at University of Northern British Columbia, we went in, we sampled, and we did extensive metagenome surveys. So we have a good idea of the general community composition of these crusts. But what we've tried to focus on is, in parallel to the metagenomics, is doing culture-based work. And so this is largely, well, for me, it was mostly Saturday morning experiments until I had a student brave enough to come in and start off something brand new. And that student is actually the student responsible for most of this work, Erin Carr, at University of Nebraska. And so she developed a workflow for analyzing the soil crust samples. So we simply grind them. Again, it's much easier to grind the crust than a rock. We plate them on a selected medium for fungi, which is rich with a cocktail of antibiotics and is also loaded with cyclohexamide. If we don't put the cyclohexamide in there, we get a lot of fastidious molds. And so then we can let these plates go for up to a month, and then we just sequentially pick colonies as they emerge on the plates. We also did a separate selection for algae as well. Then once we get the isolates, then we just do ITS sequencing to get a sense of what they are. So over several months, Aaron accumulated a large number of isolates that she classified into 6 different categories based upon their culture characteristics. And she gave them numbers and letter combinations that I could not possibly remember. So I asked her if she could, just to help me, come up with a better set of descriptors for these different strains, which she did. And so I introduce you to Goopy, which is her first one. So Goopy is a representative of one group of largely yeasts that form colonies that are, as the name implies, very goopy. They have a thick extracellular matrix. Slimy is another largely black yeast that forms colonies that are they're glistening. They're not as thick as Goopy, but they still have extracellular matrix. And then Lumpy is largely pseudohyphal and adheres to the agar very strongly, so does Dopey. And Fuzzy is mostly filamentous. And then Crusty has been the toughest one to work with. Crusty, again, these are all melanized fungi. Crusty forms desiccated, dry, powdery colonies that don't radiate out. They just grow up in themselves. It takes about a month to get a colony when you inoculate plates with Crusty. And so we profiled a variety of different phenotypes for each of these 6 fungi. And then we applied to the JGI community sequencing program, and were awarded support from JGI to do complete genome sequences for each of these 6, plus 2 representative algae, and also do all our Amplicon metagenomes from this location. So that project is still underway, and we've got 5 of the genomes. The only one we don't have yet is Crusty, and I'll tell you why we don't in just a minute. So having those genomes has then allowed us to begin to tackle some of these questions. And so the first one that I'll try to address, and I'll point out that this is all still work in progress. Because these fungi grow so slowly, it often takes a month or more to do some of the experiments, particularly the ones related to nutrient acquisition. And so most of our information on nutrient acquisition has come from our analysis of Crusty. And so this is a phylogeny of the Chaetotheriales. And so the Chaetotheriales are a family within the Urocheomycetes, and all of our isolates are members of the Chaetotheriales. And Crusty forms a unique clade that falls within a larger clade that consists mostly of Nufia species. And Nufias are known inhabitants of rock surfaces, marble statues in Europe that have been isolated from. And they characteristically form these crusty, dry, desiccated colonies. They exhibit meristematic growth, so they form uniform hyphae, so long chains of round cells. And occasionally they'll form larger cells that divide in different patterns, apparently to increase the surface to volume ratio of the fungus. And so with Crusty, Aaron made or followed up an actually initial observation that I had made. So again, these are these dry, crusty colonies. Sorry. These are these dry, crusty colonies And we had noticed over the years, whenever I was working on them, that on prolonged incubation, we'd often get these little pink colonies emerging on the plate. And I'd always assumed that this was a contaminant, perhaps a lot of red yeast contamination. But Erin noted that if she grew them on richer media as opposed to a minimal medium, we got much more robust growth. And she was skeptical of the idea that it was a contamination. And so she did a couple of follow-up experiments, and one was to simply grow in the presence of antibiotics to try and get rid of the bacteria, which got rid of the bacteria, but it basically wiped out the growth of crusty as well. And then when we submitted the crusty, very clean crusty DNA for sequencing, JGI came back and said you have a contaminant. And in fact, there were two individual peaks that both corresponded to species of bacteria. And so they argued that we had simply contaminated our DNA preps. We went back and redid all the preps, getting everything as clean and sterile as possible, and had the same result. So Erin finally followed up with some additional experiments to try and get at the issue of whether this pink bacterium, which she called Pinky, could in fact be associated with crusty, perhaps as some sort of endosymbiont. And one experiment she did was she simply verified that she could amplify 16S ribosomal DNA from crusty cells that have been exposed to a fairly harsh treatment, a bleach treatment, to strip off any associated bacteria. And certainly by culturing, they were clean crusty cells, but you could still amplify 16S. And we have some images. They're not good enough. I'm not confident enough to claim from the images that we can see the bacterium inside the crusty, the muriform cells. But clearly, there is within each muriform hyphae, there's one larger cell, and there are bacteria that appear to be moving around inside that cell. And we're just trying to deal with the various controls to eliminate other possible interpretations of that result. We do know that when we grow crusty in the absence of light, that the pinky bacterium grows quite happily. But when we grow in the presence of light, we no longer get the spread out growth of the methylobacteria species. I forgot to mention that the 16S that we amplify from crusty cells, the sequence corresponds to the exact same one that JGI pulled out from the whole genome sequence. And we've now done the genome sequence of that Methylobacter species, and it's the same one that JGI pulled out from our DNA sample. And I'll also add that when grown in the presence of light, even though you don't see the Methylobacter spread over the surface, we can see them aggregated around the black colonies of crusty. So they're there. They're more associated with the crusty than they are in the dark when they're spread out. Now, the one other thing that Erin had done was she had shown that in the absence of any carbon source, if we co-culture crusty with an alga, we mostly use chlorella, they would form these lichen-like aggregates. And we knew that Methylobacteria produce a lot of different phytohormones. And so she did an agar overlay experiment to verify that the Methylobacteria that she was isolating from crusty could actually support algal growth. And so all she's done is on these plates, she spotted colonies of the Methylobacterium and then applied an agar overlay with chlorella. And so the green that you see is the top agar, and you get robust growth of the added chlorella over top of the spots where the Methylobacterium has been added, except for one of the Methylobacterium isolates. this one down here, which was from the parking lot outside our building in Nebraska. And that particular methylbacterium actually appeared to inhibit the growth. We've made recordings from one of our study sites in Saskatchewan where the surface temperature might be as high as 45 degrees Celsius, 48 degrees Celsius in the middle of the afternoon, and it might be as low as 5 degrees Celsius 10 hours later, 12 hours later. And so, that's you have that annual diurnal or typical diurnal variation. So in order to answer those questions, we took advantage of goopy and slimy. And so these guys form, again, another unique clade that falls within the genus Exophyala, but they're a unique genus within the Exophyala. And so shown here is this is just a plate of goopy and the features of the genome sequence. So it's about a 28 megabase genome, about 11,000 genes, mostly a budding yeast. And I was going to show a movie, but it's not going to play because the movie's files are on my laptop and not Leonor's laptop. But goopy, it grows solely as a budding yeast, and it forms long chains of budding yeast, and they branch at right angles. So they form chains, monopolar chains that produce these sort of right-angled structures. On the other hand, slimy is, and actually I should back up and note that we're writing up the paper describing these fungi right now. So Aaron has gone ahead and given them proper names. Exophyala viscosium is goopy, and Exophyala lamosus is slimy. But it's hard to get away from the lab vernacular. And in fact, if you go to the JGI website, they have the genomes listed as goopy and slimy, which I wasn't very pleased about. I wasn't. And so the slimy genome is about the same size as for goopy, and the gene content is about the same. These guys are different species. They're both members of Exophyala. The growth features are different. Slimy, and again, I can't show the movie, but slimy is more filamentous than goopy. Again, it grows in a monopolar fashion, but it is much more filamentous than goopy is. But more importantly, with the help of JGI, we've done a syngeneic analysis, and while there clearly is some conservation of gene order between goopy and slimy, there are clear differences. And if we just pick random genes and do alignments, they're about 96% identical. And so we think we have enough to believe that they're separate species. Both of them reproduce only asexually. We have no evidence yet for sexual reproduction, even though the toolkit is there. So we may be able to find it at some point, and we could attempt to do crosses. But in the meantime, these are of the isolates that we've recovered from the soil crust. These are the ones that grow the fastest compared to crusty, where it takes a month to form a colony. These guys will make a colony in about 3 to 5 days. And so we've begun to ask some questions about the regulation of melanin production in these fungi, arguing that melanin is essentially the coat of armor that allows these guys to tolerate the conditions that they experience on a rock surface or in the soil crust. And I'll point out that we've recovered other isolates that appear to be, whether they're slimy or goopy, we don't know yet, but we've recovered them from rock surfaces in Newfoundland, we've recovered them from rock surfaces in Churchill, and we've recovered them in soil crusts from a number of other locations as well. So they're worldwide, or at least North American-wide in their distribution. Though I think we do have some isolates from Taiwan as well that are goopy, so they manufacture worldwide. So to understand the production of melanin, so again, melanin is this polymorphous polymer that all fungi produce. And within the black fungi, they just make it all the time, as opposed to other fungi such as Aspergillus that we had regularly worked on, where they only produce it during development, asexual or sexual development. So whenever a fungus makes a resistant structure, a sclerotium, for example, is typically melanized. What presumably allows these fungi to colonize these extreme habitats is the fact that they produce melanin all the time. We certainly see some variation in the production of melanin depending on the different types of media, so ranging from malt extract media through YPD. One thing that we noted is that in production on YPD medium, a lot of melanin is produced, but it's also secreted. So it's released into the media, and that's why you start to see this black shading as opposed to the clear colony edges on the other media. What Aaron did to get a better handle on the regulation of melanin production was to look at the impact of carbon source. So we use these, they're called C32 strips made by BioMoreau. It's the same company that makes e-tests for antifungal testing. And so each of these wells is a base growth medium, and each well has a different carbon source. And so we can assess two things here. Number one is growth, and Aaron came up with a semi-quantitative measure for doing that. The second thing we can measure is pigmentation. And so note that pigmentation varies depending on the carbon source. So in some cases, we have very similar growth. For example, Leviticus versus Mannitol. Leviticus is much darker than Mannitol. 2-ketoglutarate is also much darker than the mol, I think, was maltose. So we see a range of variations in pigmentation depending upon the carbon source, which suggests that this might provide us with a helpful clue, because what we really want to get at here is the regulation of melanin production. The actual biosynthetic pathways are fairly well understood in fungi. So there's three different types of melanin that are largely, or different classes of melanin that are made. So the DHN melanin, this group is largely the result or the product of a polyketide synthase, whereas eumelanin and other related melanins are produced through a tyrosine-mediated L-DOPA pathway. And with DOPA, it can be degraded through HGA to form pyomelanin. So there's different, structurally different types, or the melanins that are produced through different pathways. And the fact that we see this variation in melanin or pigmentation based upon carbon source suggests that there might be differential activation of these pathways depending upon a carbon source. And unfortunately, within fungi, we know very, very little about the regulation of melanin production. Again, the enzymatic pathways are fairly well understood. There's inhibitors for those pathways, but the key regulatory, the key signaling pathways that regulate melanin production remain somewhat of a mystery. So we know with our genome sequence that each of these pathways is present in both slimy and goopy. All right. Which is not always normally the case. So for example, in Aspergillus species, you'll often find that they have the DHN melanin pathway, but are missing the DOPA eumelanin pathway. A lot of basidiomycetes will have this pathway, but not this pathway. All the Chaetotheriales genomes that we currently have, they have each of these pathways. So to try and get at least an initial handle on the interplay between these pathways, Aaron has relied upon the use of inhibitors. We haven't set up we have all the tools that we need, we just haven't done the work yet to set up a gene editing platform for both goopy and slimy. So in the meantime, she's taken advantage of different inhibitors, and so far the experiments have been maybe not the most informative. So kojic acid in Aspergillus will inhibit, there are some species of Aspergilli that do have the L-DOPA pathway, not many, but in those that do have the L-DOPA pathway, kojic acid will inhibit that pathway and they become albino. But it had no effect on either goopy or slimy. On the other hand, phthalide is an inhibitor of the DHN melanin pathway, and the only effect that it had was it resulted in increased secretion of melanin, but that effect would be abolished by the addition of kojic acid. So we're seeing almost a chemical epistasis here, which suggests, at least to us, that both different types of melanin are being made. And I don't know that we can interpret any more from that at this point in time until we're able to get at it genetically. But at least hints that both are being made. And what's also interesting is that both slimy and goopy are prodigious secretors of melanin. So if Erin does a time course in either YPD or MEA medium, you can see when she spun down the cells that the medium remains quite dark, especially in YPD. And this, when she showed this at a meeting, it elicited quite a response from some of our industry partners, because there's a lot of interest in fungi that secrete melanin. It's very difficult to extract melanin. But for example, we're working right now with a Naval Research Lab. They're apparently interested in coating ships in melanin. And there's other industrial applications as well. And there aren't many yeasts that actually secrete the amount of melanin that we get from our slimy and goopy species. And not only that, we apparently can see different types of melanin being secreted. So there are different extraction protocols for melanin. There's a relatively mild enzymatic protocol that's focused on denaturing proteins, and a much harsher protocol where you use alternating acid-base treatments to just strip off the melanin. This is typically used for extracting melanin from within cells. And so Erin applied this to cells from supernatants from cells grown in YPD or MEA. So the melanin that was secreted in MEA was sensitive to enzymatic extraction, whereas the melanin that was secreted in YPD was resistant. So they behave differently. So it appears that there are different melanins being made on those, or being secreted on those two different types of media. On the other hand, if she does the chemical extraction, then all the melanins are extracted. They form a variety of different shapes of polymers, and she's doing some experiments right now to see how well she can resuspend them. And then the very last thing is we've also taken advantage of the genome sequences to begin doing some genetics. And so Erin has used EMS mutagenesis on both goopy and slimy to simply select for mutants that have altered pigmentation. So in some cases they're still melanized, but the melanin is being dumped out into the medium. In other cases they're no longer melanized. So this is the only albino, so to speak, mutant that she's made. It's pink, not because it has an endosymbiont. It's pink because of the presence of carotenoids. So besides making melanin, these guys also produce a variety of carotenoids. And I can give you a separate talk on what we know about the regulation of carotenoid production in these fungi. So even if they don't have melanin, they have a backup system. And in fact, when we resequence the genome of this mutant, it has a nonsense mutation in the PKS that produces DHN melanin. And one of my students who's still up at University of Manitoba is doing a similar screen with a different Exophyalus species, and he's generated about 50 different mutants that have either they're albino or they're hyperpigmented. And so far, every albino mutant that we've found has a mutation in PKS1, which leads to a really puzzling question, because I told you earlier that these guys can make both DHN melanin and DOPA melanin. So the mutations in PKS1 will eliminate DHN melanin. The DOPA melanin should still be there. So why are they albino? We don't have an answer to that question yet, but we do know from the work up at Manitoba that half of our albinos up there are conditional albinos. Depending on media conditions, we can turn them back to being melanized, whereas some of the others can't. They all have the same set of mutations in PKS1, so we're looking at the other mutations that distinguish those mutants to see if we can get a sense of why some are conditional and some aren't. And then one other direction that we're hoping to go with this is to integrate modeling. So one of my colleagues at Nebraska, Rajiv Saha, he's an expert in metabolic modeling. And so his former student, who was actually former alumnus of Iowa State, Wheaton Schroeder, developed a metabolic model to help us answer the question. The hypothesis is that the production of melanin, and perhaps carotenoids as well, would impose a severe metabolic cost on these cells. And this might be why they grow so slowly, for example, because all the carbon is being funneled into melanin production. And the results from the model, building the model was a challenge because he did not have a lot of genomes to work on at the time. But the model suggests that there is a penalty, but it turns out the penalty is greater for making carotenoids than it is for making melanins. And so we're hoping to build off of this model by integrating some of the data from Goopy and Slimy to refine that and get a further sense of where the penalties are exerted in terms of producing melanin and carotenoids. And these models will also help us to predict and validate some of the regulatory pathways that we're interested in. So just to finish up 2 final slides. One is answers to the questions. So the take-home message. So how do these extremotolerant fungi adapt to extreme environments? Not a huge surprise, it's the melanin, right? And I didn't mention it, for example, but in the albino mutants that we do have, they are seriously compromised for the ability to survive UV, for example. And if we impose other stresses, they're much, much more sensitive than wild type. And then the tougher question that we said there's still a lot of loose ends is the idea that endosymbiosis or perhaps transient lichenization are important for nutrient acquisition in these habitats. And then the last slide is where do I want to go with this? So some of you here, I know Leonore and Allison and Mark and Navi, I've been bugging you guys for isolates of various fungal plant pathogens. And what these pathogens all share in common is that they're highly melanized. So Macrophamina, Cellosphaeria, the different pseudoblock pathogens. And so we're interested in extending some of this work so if we can get a foothold on the regulation of melanin synthesis in our extremotolerant fungi, we might be able to leverage that to look at how inhibiting the production of melanin might mitigate the capacity of these fungi to overwinter. My argument is that overwintering is an extremotolerant environment. Maybe not so much now as it used to be, but the ability to overwinter in crop residue presumably requires some of the same toolkit that our rock-inhabiting fungi, our biocrust fungi are using. So if we can compromise that toolkit, that might be a way to reducing propagule formation once you emerge out of the winter. So initially, we've just been doing some genome annotations on these fungi. All the melanin production pathways are there. And then the next step with both Macrophamina and Cellosphaeria isolates is we want to start looking at variation in response to different carbon sources, just to see if we can alter pigment production by virtue of different carbon sources. And then last, just the people who did the work and supported the work. Now, just the people highlighted in yellow are the students at Manitoba and Nebraska who directly contributed to this work. That Erin is my last PhD student at University of Nebraska, and she's a superstar, and she'll be defending early next year. And most of this work is the result of her effort. And I'll stop there, and thank you for listening.